what we're doing is that what I'm doing here is just adding on a few extra things so that I'm sure that you're going along the right track. First one just to talk to you about is downloading the software itself. These are Java-based programs, and if you're using your own computer rather than a university computer, then you may be required just to download Java before you can use them. But having done that, you should be able to go to Google and find and search for Artemis comparison tool or Artemis on the Sanger website, and go to the tool pages. If you found one of those tools, then you can go from there and have a look at getting the other software from the same site. Artemis itself is described as being it's described as being an annotation piece of software, and this is what the lecture is about, this idea of being able to look at DNA and look at the evidence around it for inferring where genes are, changing gene models, and collating evidence around those gene models. So you'll notice that there are three options that you can pick in terms of operating system: Unix, Mac OS, and Windows. Please pick the one that's appropriate for your platform. In this case, I'm using Windows here for this demonstration, and so I can click on this and it will download to my desktop with a download file. You might notice that in some versions of your browser it might have problems with FTP, and I found that particularly for this version of Explorer, which Windows like, but you can swap over and use Chrome, and that overcomes some of those problems that I've found there are with Windows Explorer and FTP. So I apologise for my cough, but what we can then do is then go over to the course itself, go to module content, and to the section where my lectures are, and then you can download the files themselves for the Artemis workshop. Okay. Now you'll also notice that when you open these, they will download. They will download those files, and you can open them. Well, you should save them and then view where the download is. One of the things I want you to be aware of is that there's this sort of interesting thing in Windows in particular, which it doesn't happen in the Mac, is if I double click on a zip file, then I can go in and I can use those files. However, Artemis isn't able to look at the files inside that zipped container until you've actually extracted all. So when you extract all, you can choose where they're extracted to, and once those files are extracted, then you will be able to use those files, and they will be available for Artemis. The data there, just to so we know what we're doing, and I'll move over the zip again and just remind you that we need to extract that zip before Artemis can see it, and we'll do it there again onto the desktop itself. So what we've then got is we've then got a folder in which the data is contained, and it's worth just having a quick look at some of that data and thinking about some of the formats that were involved, because the formats that you're seeing are one of two formats. There's either a FASTA file, which can be looked at in something like Notepad, and we can get Notepad up here, And if we open this, we'll see that it's just a text file. Again, going to the desktop where things are. Now, you'll see that the obviously, because this is not a .text file, we need to just get that off. We can have a look at this, and we can see that this is a standard format FASTA. Okay, and this can be opened by Artemis. And if we go and have a look at the other file type that's in other file types that are in here, we can open those up, and we can see that they are all EMBL file, which is directly from the database, and this is a EMBL file. The format from NCBI is very similar, but what this does is it gives you a list of those features. So you can see wherever it's got something like CDS, it's followed by a coordinate of the location of that gene or feature, and then some information about how that feature should be interpreted, or special information about how you can arrive at getting the protein from that, so the translation table that should be used. It's got the name of the gene, the function of that, and the product of that particular region of the genome. Also, database accessions and things are also available within this bit of the file. So all of these files are human readable, and you can interpret them and you can change them in a text editor or in a in just a simple text editor like Notepad. You notice also there's some tab files, and they're just a reduced version of the EMBL file that we were looking at, which Artemis can load up and interpret. We'll also use in the Artemis comparison tool later a crunch file, and it's just worth having a quick look at those in Notepad, because they are themselves just columns of numbers, and although they haven't quite translated here, there's a tab delimited file, and it just contains the coordinates over which the matches occur between these two genomes. So there really isn't anything sort of overly complicated about these files, but Artemis can load them in and interpret them, and importantly, draw some graphics on them. And when we double click on the Artemis icon, if Java is installed, then you should see this little coffee mug there steaming as part of the Artemis icon. As I said previously, I've set the working directory to be the desktop, and so when I click OK, then I get this window here, which is the sort of Artemis main sort of program front window. Now from here we can open a file, we can set various options like the genetic code tables, and we can get some information about Artemis itself. But we're just going to go straight and open a file. Remember that if we want to see inside these files and see all of these files, then we need to ask for all files to be shown. And on this occasion, we're just going to load in the EMBL file to start with, open that up, say no to the warning on this occasion. Then we can see that we've got this familiar window, and as I was saying previously, we can see that this window has the interactions, or the sort of the menus up above here, which can also be got by right-clicking on specific areas as well, remember. But we have these menus here. Then we have the entries, which are currently being viewed in Artemis, and remember that we need to have a yellow box around here so that this is the default entry. If we want to change the default entry, one of the things we can do is we can either read in a new entry, read an entry, or we can create one. So if we come to create, we can come down and say create a new entry, and in doing that we create one that says no name here. If we want to change the name of that, then we save an entry and I select no name, and I can just save this as, I don't know, stuff or my stuff, and then save that, and then you see it's changed that name to my stuff. Now I can actually, if I want things to go into my stuff that I create in here and not into the Emble one here, what we can actually do is change the entry. So we can go down and then select the default entry as being my stuff, and you notice that the yellow box changes over and becomes my stuff as the entry. Okay, so if I click off the Emble entry, you can see that my stuff is empty because there's nothing listed here and there are no boxes there. But I can change that quite easily because I can select a piece of DNA here. I can create a feature from from the base range selected, and then I'm going to put this in as a miscellaneous product, as a miscellaneous feature, and apply that, and you can see the green box jumps from the frames onto the DNA sequence itself, and say OK, and then that's there. So if I click off that, you can see that it disappears from down here, and if I click back onto the entries, you can see that that the other features come back and this one remains here. Now if I click on this now, then this feature will appear at the bottom of the features in the rest of this genome. But if I want to find it again, then I can just click off and go back to it. So it's important to remember that we can edit this feature by pressing E, and this allows us to see that miscellaneous feature, and we can change various things associated with that, particularly the coordinates. We can add a qualifier, a note. We say this is a random box, okay, because it is. We can apply that, and now when we look at that, we can see that this is a random box is shown in this descriptor window here. So what we've got then is control over the things that we can put in, the things that we can modify on here. And one of the things that we can also do as a sort of an advanced tool is select a gene, and then we can move it to a different entry, or we can copy it to a different entry. So I can copy this gene that I have selected here at the moment into my entry, and when I click off, you can see that I've got it here. And then I can do various things to change this. I could trim it to the next methionine, and that will change in my view here, but won't change in the original entry. Okay, so these sort of having multiple entries is quite a nice way of sort of keeping your own notes on the genome, or changing genes that you want to change just for experimental purposes. So let's put the full data back again, and just turn off my stuff for the minute, because I want to start to have a look at some of the features that are in a genome. So if I click on any gene then and double click on it, what will happen is that it will bring that gene into the center. And remember, the way that it's set up at the moment, we can see the amino acid translation for that region here in this lower region. In selecting it, obviously, and then pressing E, we can see more of the details about that gene, and this is exactly the same details that are seen in the EMBL format that we looked at in Notepad earlier. So this is one way of getting to that code that is there defining the features and properties of that gene. It's also important to realize that when you do that, you notice that you've got these things like key, which is a CDS or coding domain region, and you've got qualifiers, which are basically all of these notes that are down here. So we can use the keys and the qualifiers to search. So when we want to navigate through the genome, we can go to select and we can look at the feature selector. And here we can use things like key to search for. So I can go and have a look for, say, RBS binding sites or maybe CDSs. And I can go through that and I can search the notes of the qualifier, and I could look for something, say, like toxin. I'm going to check that I've got it case insensitive here, just in case toxin is spelled with a small t somewhere in this genome, and I can select that one through up to where the next gene is that might have a note saying toxin, and I can view it. And on this occasion there aren't any, but I could have a look at something like sigma factor. I could select those, and then I could view that entry that has a sigma factor here. Now, one of the reasons is that you'll notice is that if I put toxin in here and I look at note, I might not get an entry, and when I view that, I haven't got anything to look at. If I change this to qualifier, or if I change this to product, then I might get a slightly different answer. So I put this in and select, and then view, and you can see on this occasion I have two annotations in the qualifier that contain the word toxin. Okay, so this just shows you as well that sometimes actually searching by qualifier can be quite difficult. So if we can click off that and ask the question, how many are there that have toxin in the key, then we can get the same answer. Okay, let's pick a sort of a better example of that and say type, and we can select those and view, and you can see that we have a lot of things that are selected that have type in their annotation. If we have a look in the product, we will see that type isn't always described within the product code. Sometimes it's going to be shown within the wider qualifiers themselves. So we can actually look at the wider qualifiers by right-clicking and having a look to see where that, say, type annotation had come from. One of the things, obviously, right-clicking and changing some of these things like gene name that I can see here is a very useful thing to be able to do. You'll notice also that we have a sort of 111 selected features that have type somewhere in their qualifier, which again is a shortcut to understanding how many genes there are. It's worth saying that with all of these things, when we have them selected, a list selected, we can carry out operations on this list of selected items. So we can go and do this feature search again. We can view selected features, and we can get an overview of those selected features. So for example, we've got 111 counts here of CDSs, and it gives us a rundown on what the makeup of those are. You know, how many what's the sort of the GC content, how many partial counts are there, how many splice CDSs are there, so on and so forth. So it's worth remembering that this view window does give you that sort of extra control over the things that you can observe. But if we go back to the main list again, if we're looking for overall information, then the overview can be found in view. And again, if we click on this, then it gives us a genome level view of what's here, the number of bases that there are in the genome, the number of CDSs that there are in the region, and gives us that detail in a bit more fine detail as well. It gives us the number of miscellaneous RNA sequences, number of RNA sequences, the number of tRNA sequences, and so on and so forth. How many promoters are there? How many repeat regions? And this last one here is particularly useful. This is a summary of active entries, because it actually gives you a list of all the different keys that are in use in the genome as well. So if we come back and we want to select things by key, then we can do that. We can say, right, okay, we remember from that we had a number of ribosomal proteins. So let's go and find ribosomal RNA genes. We can go through and we can find those, and we can see that we've selected all 22. Now, if I want to view those, then a good way of doing that is to go to feature filters and then go to selected features in that submenu there. And then that gives us the same overview that we had previously. In other words, these little summary lists which allow us to go down through and have a look at them at our leisure. Obviously, showing you how powerful some of these sublists can be, because what we can also do is click on any one of these, and it will actually take the product list down here to that region. But if I double click, it will center the Artemis view as well on that gene that I'm clicking on. And this is incredibly helpful, because that means we can make these lists, selective lists, and then go through the genome with the cursor and actually find out where those different genes are, and look at their location, and take some notes, say for example about the genes that we're finding around these particular sets of genes or types of genes that we've selected. So that's a kind of, again, just a reinforcement of selecting genes and having a look for them within Artemis. One of the things you'll also know is that there's the navigator window, which is very powerful, allows you to go to a particular base. So if we put in base 1 million and then we say go, then it will take us to base 1 million. Okay, we can also do the same if we know what the gene name is, or if there's something we want to find in the qualifier, then we can go straight to that. Now the difference with this is that it doesn't give us a list of genes. It will just take us to the next annotation that has a toxin talked about in it. But it's also this window that we can use to find the next repeat. For example, we go to, okay, it's not found that, a repeat region, and we can go to the next repeat region and it will find that for us in the genome. But again, notice it's not generating a list of all of the repeat regions in the genome. We can also go and find particular base patterns that are within the genome, or particular peptides that sequences that we might be interested in. But remember again that it's not necessarily going from the beginning unless you force it to go from the beginning, and otherwise, or you can go from the current location which is selected. So just remember as well that when you select a feature like this repeat region here, when you look at it in this window, it's giving you the key first of all, or the gene name depending on how you have this selected. Then it's giving you the start and the stop coordinate, and then the C here means that it's on the complementary strand. But remember that you've got this information here for knowing the number of genes in the region that you're in. So one of the things that can be a little bit difficult sometimes to do in Artemis is to find a larger region and to define that larger region and keep hold of it. So my sort of trick for doing this is that I make sure I've got a default entry open, and then I select just a random piece of DNA and then say create feature from that selected piece of DNA. Now what this allows me to do, as I showed you previously, is to just make a change the key. Normally I do to miscellaneous feature. I might add a note and say region 1 test, and then I can apply that, and you see I get this green feature on the DNA in the area that I previously selected. But say that I want to go to a specific region, and this one is one which is at 20,000 bases and is goes to 40,000 bases. Then obviously I need to make sure I get the syntax right, but I can apply that, and now that key, that feature is now associated with this region. So I meant to say 20. There we go. So I can change that when I realize I've made a mistake. I can apply that, and if I click on that, then I go to it. If I take off the rest of my features, I can go and find it in that region, and I can zoom out as well so that I can see the whole region again. So now I've got this region, I can find a specified region within this genome and work on that by itself. And I think that this is one of the more efficient ways of actually finding a specific region and then keeping it in an active entry so that I can go back to it and find it again. So when we've finished working on Artemis, we can go back and save the entry. We can save the entry my stuff, and then we can use that for later on, and then we can just close it down. It says, are you sure there are unsaved changes? If you're really sure, then just answer yes and it will close up. So interestingly, it saved my stuff, the entry as a Excel file. I haven't got a clue whether or not that has worked and if it really is an Excel file, but it should be a tab delimited file in the current format that it's saved in. And of course that's been saved to the desktop here so that you can look at it. So what I want to do next is go on and open up ACT, and ACT opens in a very similar way, but you get a slightly different options associated when you want to open it. So when I open this, now what I get is I get this this view where I can enter multiple sequences, but we're expecting that. And for this demonstration, I'm just going to use the DNA from going to use the DNA from the E. coli. Actually, sorry, as I'm going to put the DNA in from the Salmonella as sequence 1. Then I've got the comparison file, the crunch file between the two. And then I'm going to open up the E. coli as the second piece of DNA. In this case, actually, I can use the EMBL file here and use that. And that has the advantage of that it has the genes on it already annotated on. Whereas with the Salmonella gene, the DNA, they won't be obviously already there, but we can add those, and I'll show you how to do that in a moment. So we click apply, we ignore the warnings for the moment, then we should get this, this view again. What we have really are two of the sort of 6-frame Artemis views, one for genome 1, which is Salmonella, and the second for genome 2, which is E. coli. And then these red lines are the regions that show homology between these genomes according to BLAST. And also, as I click on them, they turn yellow. If I double click on a region, then it will center itself. Now, I'm going to zoom out using the top one as the example, and what you'll be able to see is there's lots and lots of thin, fine matches that go between them. So it's important to sort of be able to filter those out, and I can do that in two ways. I can either use this slider to filter out so I only get the best matches, or as we're shown in the workshop, you can then go in and and actually set a cutoff score and then work out where you want those matches to be. The two ways of doing this are very similar. The advantage of using the slider is that you have a bit more visual control over what's happening. But as you zoom out, again, things become a bit wonky sometimes, so just give it a double click and it will bring things back into the center. So the red lines show homology, and so do the blue lines, but in this case the blue lines are showing where the DNA is reverse complemented between the two genomes. There's been an inversion between the two genomes. So it's always nice to zoom out and have a look at what's happening in the genomes to give you sort of an overview, but then we kind of want to go back in and have a look at some of the nitty-gritty of what's actually going on. So as we come down into this, we can begin to see these regions which are shared quite well between them. And I'm just going to increase this a little bit because it doesn't matter quite so much if it's a bit noisier down at this level. But you can see I probably want to just try and keep some of these big blocks without keeping these little lines, but getting rid of the little lines. And what can begin to see are these characteristic V patterns between the two genomes, where we've got regions that are novel to this top genome because they're not in this genome below. We can see that in this genome here we have a region that isn't in this genome, but has been inserted, but there's been an insertion in this bottom genome. And because we have the E. coli genes in already, we can see what those genes are that have been inserted in this region. So this sort of white triangles are very characteristic of genes being inserted or deleted between the genomes. We can, again, just as we did in Artemis, load in annotations. So if I go to File, then I go to the genome which I want, which is number 2, I can read an entry into genome number 2, and in this case it's the tab data. So if I add that, open that in, I ignore the warnings again, then suddenly I can see the genes have appeared, and now those regions which are novel to the Salmonella have annotation relative to E. coli. In a perfect world, if we look at these two annotations between these two genomes, by clicking E, we should be able to see that they're the same gene, and in most cases that will be the case. So the Artemis comparison tool gives this really nice way of just sort of going in and doing quick comparisons between genomes. We can see that we've got these blocks of homology which allow us to say that these genes in this genome are likely to be the same as these genes in this genome. It also helps us find these regions of novelty between the two genomes. We can also select things in exactly the same way on either one of the features that we did in Artemis. So we can select things by key, for example. We can select all miscellaneous features as we go through, and all of those miscellaneous features that have homology, an area of homology overlapping with them, will also be selected. And we can view those in the same way that we did previously by going to, by forming a list, by going to feature selector and select function, select selected features, and we get one of these lists again that we got previously. Remembering that this is the list of, in this case, miscellaneous features that are in Salmonella. Again, we can perform the basic stats on those by going to View and having a look at the overview. There are some novel things that we can do. When we're looking Artemis, Artemis comparison tool, if I click on this window, then you can see that one of the features I have is to select various features, but I can select all features, sorry, the mouse is jumping around, all features in a non-matching region. And if I do that, I say yes, I want strict overlap. And you see those novel regions in Salmonella are being selected versus those that are in the conserved region with Salmonella and E. coli. So this is a nice way again of picking out the novel regions, and again I can make these into a list by going to View, picking the right genome, feature filters, select features, and then having a nice list of genes here. Okay, and again I can get an overview of those by going to the overview. Remembering that this tells me not just CDSs, which are details there, but also things about the miscellaneous features and the number of repeat regions. It should be noted that this data is a summary for the whole of the genome, not just for the regions that we're just looking at just now. If we wanted to do that, then we can make a private list by selecting these again. So just in the same way that we did previously, it's possible to use the go to navigator function to go to regions in the genome, go to a particular base within the genome, and it's also possible to search on qualifiers and features. And remember, if you remember which genome you're searching in, if you're searching in the top one, Salmonella, if you're searching in sequence 2, then it's E. coli in this example, and allows you to switch between those two. It is possible again to do things like at the moment, if I move this cursor along the top, then you'll notice it's all fixed and it moves with me. Sometimes this causes some problems and makes it difficult to align up. So for example here, you can see that this is all a bit sort of wonky and it's difficult to get it all in the same window. So I can actually unlock these sequences, and then that means that these move or slide independently. And sometimes this is helpful because it just allows you to align things up a little bit better. But remember to lock it back, because sometimes it can be really difficult to work with these independently moving sequences. Again, remember, you can save everything out, and you can save them by features. You can take and you can print things to screen or save screenshots through using the file manager as well. Remember again, of course, that you can add graphs for GC content or deviation, and those can be added and can be done for both sequences that you're looking at. So although things can be quite straightforward to look in the ACT comparison tool, there are some features where I will go back and use Artemis. So for example, if I know now the region I'm looking at and I want to look at those genes in more detail, sometimes it's easier to do that in Artemis. And if you go and have a look at that, so for the TIFI data, I can actually say edit in Artemis, and it will open the genome up in Artemis and allow me to look at the genome in Artemis by itself. And then when I close this, some of the changes will be then found back on this genome if I've made any. So you can switch quite easily between Artemis and ACT, and sometimes it's as well to do that because some things are just easier to do in the Artemis windows.